The overall goal of the following experiment is to generate predictive models for transient expression of proteins in plant leaves. This is achieved by setting up an experimental design to identify the most important parameters for transient expression and quantify their impact on protein accumulation. As a second step, genes are cloned and transferred into agrobacteria, which are then injected into leaves, resulting in transient protein expression. Next, leaf samples are analyzed in order to determine protein expression levels. Results are obtained that show the pattern of transient protein expression levels in leaves of different ages and for different incubation conditions based on a design of experiment model evaluation.
The main advantage of this technique over existing methods like one factor at a time is that interactions between different parameters like leaf age or position on leaf can be detected and quantified.
The one factor at a time approach that is often used to characterize the effect of certain factors on the outcome of an experiment is suboptimal, because the individual runs during an experiment will be aligned like pearls on a string, thus achieving a low coverage of the design space. In contrast, with the design of experiments of DOE strategy, the variation of more than one factor at a time enhances the coverage and thus the precision of the resulting models. Furthermore, the biased design space coverage in one factor at a time experiments can also fail to identify